Hi, everybody, and welcome to the Student Research Symposium for 2021. We have a great list of speakers, and I'm excited to hear from our speakers this afternoon. Unfortunately, we can't be in person, but this will still be another excellent venue for presenting research, learning a little bit more about the research that's done here at TCU. So without further ado, we now present Emily Burnham, and she will speak on warm or cold dark matter, a love-heat relationship. If you have questions, please put them in the chat. Only the host will see those questions.
Ken, can you make the presenter co-host?
All right, I think I'm unmuted now. I just need to have screen sharing enabled. There it goes, perfect. All right, can everybody see that? Perfect. Well, good afternoon, everyone. My name is Emily Burnham, and I'll be presenting my research on dark matter, titled Warm or Cold Dark Matter, a Love-Heat Relationship. And this is advised by Dr. Mia Bobo. I'm also a part of the Department of Physics and Astronomy, and I'm really excited to be able to talk to you guys today. So you're probably all asking yourselves, well, what is dark matter? Well, in short, only 5% of the universe's total mass is 5% matter. Regular matter being baryonic matter. That means stars, planets, galaxies, comets, you and me, that's baryonic matter. However, 27% is dark matter. But what makes dark matter so special, and why is there so much of it? Well, the short answer is we don't really know, and that's why there's so much research about dark matter. Initially, it was discovered in 1933 by scientists studying the rotation rates of galaxies and seeing that they're measuring a lot more mass than they're observing. And so they titled this missing mass dark matter. Dark matter is essentially invisible and cannot interact with light. It does, however, interact gravitationally with baryonic matter, and that's how we're able to study it today. And so since it's able to attract other particles gravitationally, it creates what we call potential wells. Potential wells call gas and dust, and they all infall, and that's what creates galaxies and stars and everything else around us. And so why do we care if it's warm or cold? So if you think about particles, the hotter it is, the more energy it's going to have, and the less it's want to interact with other particles. The colder it is, it's more lethargic, and it wants to relax, and it wants to infall with other particles. So if you look on the right, these are some simulations done on how cold dark matter and warm dark matter affect regular baryonic matter. And so if you look on the top right diagram, you see cold dark matter, and you see it has lots of little pieces and little areas that are infalling together, while the warm dark matter is a lot more spread out and smooth. That's because stuff isn't falling in with each other. And so if we examine really ancient galaxies, we're able to discover, is our universe a place of warm dark matter or cold dark matter? And this brings us into my research very specifically, is we're asking the question, when did the first stars ignite and when did the first galaxies form? So we are running simulations based off of warm and cold dark matter universes. And we're examining these dark matter halos and how they behave with their masses, how far away they are from our universe, and basically how many stars that they're forming. And so we're running these simulations and we're comparing warm and cold dark matter. However, how do we know if we're right? Well, we won't really know until we have giant high dispersion telescopes being launched into space so we can probe extremely deep into space. And so if you look on the right, we have the James Webb telescope launching on Halloween of this year, and the Nancy Grace Roman telescope launching about the mid-2020s. And so these are some of the plots that I have created to understand why is this happening. And so on the left, you have mass versus distance. And you can tell that the farther you look away from the Earth, the smaller these dark matter halos are. That means that these galaxies are going to be more ancient with older and older stars. So if we look to the diagram on the right, this is the number of halos that are forming stars at a specific time. So we have a first population. This is about 20 million years ago. We're looking back, and there's a first population and then a second population over at about 60 million years. This second population is what we're assuming when the first stars formed with cold dark matter. This simulation was one on only cold matter, but if we go on to warm matter, we expect this second population to completely disappear. And so moving forward with our research, right now we're only doing very simple models based off of semi-analytic model simulations of these really complex simulations based off of dark matter halos. But eventually we plan to invest in large supercomputers who are able to do all of this thinking for us, and then do the same comparisons with warm and cold dark matter. And then finally, as soon as James Webb and Roman launch, we'll be able to see if we were right and determine whether or not our universe is a warm or a cold dark matter universe. And with that, I think I will take questions. Thank you.
So you may place questions in the chat, and I believe you also have the function to raise your hand, and we will try to monitor the room for raised hands as well. So I know you said there's sort of a difference in what you expect in terms of hot versus cold dark matter over time. Can you elaborate a little bit on the different time course in terms of when you expect to see?
Give me one, sorry, my thing's all freaking out. I'll go to here. And so I'm assuming you're going to be talking about these graphs. So when we're looking, so we have to remember that these distances that we're looking at, they are humongous, millions and millions of light years. And if we remember that light travels at a constant speed, that means it takes time to travel from one distance to another. So essentially, the farther you look away from the Earth, you're essentially probing deeper into time, and you're looking back as to when that light first is hitting you. So the deeper that we look, we're able to study, oh, if we look far enough, we'll be able to see when these first stars formed. However, it depends on whether it was warm or cold dark matter. If it was cold dark matter, it's super far out, and we have to invest in a lot of materials and able to probe the universe that far. If it's warm, however, it's going to be a lot closer than we think it is, because everything formed a lot later. Because remember, warm particles don't want to fall in with each other.
Thank you. All right. Let's see. We are going to take a second to switch hosts. Thank you very much, Emily. Appreciate it.
Thank you.
We now present Charles King, who will speak on GIS-based evaluation of the effectiveness of trees to mitigate urban heat island effects in Fort Worth, Texas. Again, if you have questions, you may raise your hand or place them in the chat. Only the host will see them, and we can then ask them during the Q&A period.
Okay, hope everyone's doing well. Like Dr. Lysing said, I'll be doing a GIS-based evaluation of the effectiveness of trees to mitigate urban heat island effects in Fort Worth, Texas. So a little bit of background: the urban heat island effect is characterized by increased temperatures in urban areas due to buildings and other great infrastructure. These buildings and other great infrastructure are better at absorbing and maintaining the heat that's placed on them. And so they are differentially heated to a higher degree. And this has a large impact on everything from human health to energy usage, soil ecology, and urban ecosystem functions. And one fantastic proposed mitigation strategy is to use trees. This is because trees provide cooling through the attenuation of thermal radiation, basically capturing the thermal radiation with their leaves, and then cooling with evapotranspiration, which is a process of taking water up through the roots, through the stem, and then out through the leaves, taking the excess heat with it in the form of water vapor. So the objective of this study was to assess if trees are a useful mitigation strategy for Fort Worth specifically, and to see how it affects the urban ecological setting. So first I had to define what my study areas were, and my target areas for the analysis were large paved areas that have a low building density. And this is for two main reasons. The first being that trees provide more cooling when they're closely associated, and it also allows for a greater ability to convert that paved surface into grass. And 4 of these areas were selected for analysis. But looking deeper into the reasoning behind this, look at a paper by Font et al. In 2015, and this compared the spatial arrangement of trees and grass to land surface temperature using a statistical analysis known as local Moran's spatial correlate. And this is basically saying that as a pattern of objects are more clustered, the Moran's value approaches 1, and as they're more dispersed, they approach -1. And if we look towards the bottom graphs, going to be focused more on A and C because those are during summertime temperatures. And as we can see, as the Moran's value approaches 1, the objects are more closely correlated. We see that the land surface temperature decreases. So it is in our best interest to make sure that when we plant these trees, we have enough room to have them closely grouped together to provide the best cooling effect. So here is a map of the 4 areas I chose across the 4th area. And in order to start making a map of our original land surface temperature, I took Landsat 8 data, and all this flow chart is telling us is that we're relating the intensity at which electromagnetic radiation reflecting back off the Earth, we're correlating the intensity at which it reflects back to how hot it is on the surface. And so by doing that, we can form a map, and this is a map of Fort Worth and the land surface temperature over the summer months. We can see that in some areas where it's cool, we're at 32 degrees Celsius or around 90 degrees. And in the hottest areas, it's 44 degrees Celsius or in excess of 110 degrees Fahrenheit. And so in order to project a map of the cooling, I first had to categorize what ecoregion Fort Worth is in, which is a cross timber ecoregion. And then I had to choose a tree that was best suited for that region. And that would be the Texas live oak. It's popular because it is hardy and it can resist drought conditions. It has a broad canopy, and that broad canopy can attenuate solar radiation very well. And previous studies have shown that it can cool a given area by 1 to 3 degrees Celsius. And so in order to project this, I took my study areas and took the middle of the study area and cooled it to 3 degrees Celsius. And then spreading outwards towards the edges, I cooled them to 1 degree Celsius. And so as we can see, this is the projected map, and it's better viewed when you can see them side by side. So as you can see, the study areas that we chose ended up being some of the hotter areas in Fort Worth, but when projected with the cooling effect, we can see that we have a significant effect on the daytime temperature there. And so why is this important? Obviously, this is important for human health, as no one likes to live in temperatures of excess of 110 degrees Fahrenheit. It's extremely uncomfortable, but it also has an extremely large ecological effect. Increased soil temperatures lead to increased rates of organic matter decomposition and nutrient cycling, and it can also explain up to 75% of soil respiration. And both of those are extremely important in the liberation of carbon from the soil into the atmosphere. And it was previously thought that under paved areas and buildings, the soil was largely inactive, but this is actually completely untrue. And 2 papers published by Edmondson et al. In 2014 and 2016, the top graph shows that in urban settings, soil organic carbon is higher in urban areas than in non-urban counterparts. And this is especially true in association with woody plants. And the density of that soil organic carbon, as shown in the bottom chart, shows no significant difference in densities, paved and unpaved surfaces. And this is extremely important because if that soil organic carbon is broken down and liberated and put back into the atmosphere, that means more CO2 is being put back into the atmosphere. And as we know, CO2 is a very potent greenhouse gas. And this could actually produce a positive feedback loop and continually heat the city to higher and higher temperatures. So while this is why trees are such a great way to mitigate these effects, because trees not only provide shade and cooling that can stop the decomposition of below ground carbon, but they can also act as a sink for above ground carbon. And in other unrelated things to soil, it can alter plant and animal phenology. Warmer temperatures can lead to a 20% increase in growing seasons, as well as influence differences in the migration patterns of birds. And in terms of human effects, decreasing urban summertime temperatures can reduce the energy usage by air conditioning by 2 to 4% for every degree Celsius we're able to decrease the temperature. And this is important as urban areas emit 71% of energy-related CO2 emissions. So in conclusion, to wrap this thing all up, planting trees can be a useful mitigation strategy for urban heat island effects, but only in the correct context, as we saw with these large unpaved or these large paved surfaces with low building density, so we can correlate the trees closely together. And lowering temperatures is in our best interest because we can reduce the energy usage, help human health complications, and stabilize the ecosystem interactions. But if we don't act quick enough, the temperatures continue to rise, the emissions of carbon from urban soils may compound the microclimatic heating of urban areas in a positive feedback loop. And so moving forward, a true multi-year study that investigates this project using in situ measurements is warranted to understand the true effect that trees would have both in the cooling of the area as the soil interactions as well. And these could look at different things such as the rates of mineralization, microbial responses, and other ecological factors that are related to temperature differences. And that is it. Thank you for listening, and I'll take any questions.
Unfortunately, we don't have time for questions at this time. So if you do have questions, then please email Charles directly or find him afterwards in potentially a Zoom room. All right, thank you, Charles. I appreciate it. We now present Riley Parades, who will speak on functionalization of pistachio biochar for nitrate removal in water.
Thank you. Okay, hello everybody. Thank you for listening and tuning in today. I'm excited to talk to you a little bit about my research. I was able to perform research on the functionalization of pistachio biochar for nitrate removal in water under Dr. Omar Harvey. So nitrate is commonly found in rivers, lakes, and groundwater. The primary source of nitrate infiltration into these groundwaters is agricultural inputs, the first and foremost being synthetic fertilizers, often highly contaminated with nitrate, or I should say they contain lots of nitrate, and then also manure application, which is a natural form of fertilizer. The EPA limits nitrate concentrations in public waterways to 10 milligrams per liter of nitrate N. And the effect of exceeding amounts of nitrate in water has devastating effects on human health. The foremost effect is that it interferes with hemoglobin's ability to transport oxygen. And this can manifest in a syndrome called blue baby syndrome, which actually causes young children to turn blue due to a lack of oxygen to their tissues. So while we wish this wasn't an issue, actually 17% of the United States groundwater is considered contaminated with nitrate. So I wanted to come together with this problem that is largely caused by agriculture and try to look into this agriculture for a solution. So my proposal was to find a large biomass of agricultural waste. And with that, I looked at my home state, California, where 99% of pistachios are produced. Pistachios require lots of nitrate to grow. And so they're right over also a lot of groundwater stores, and this causes a lot of contamination. So using the problem as a solution was my goal. So I wanted to take this pistachio waste, which has no current use, and transform it into a charcoal material that then could be used to act as a water filter for nitrate. And so the objective of my experiment was to determine the most effective charcoal at removing nitrate. And so what I did is I created 4 different biochars. The first of which was a 400-degree sample that I left at 400 degrees for no time. A second that I had heated up at 400 degrees and I maintained that for 10 minutes. And then I did this again at 600 degrees. So one sample was held for no time and one sample was held at 600 degrees for 10 minutes. I then wanted to go and see the reactivity of these biochars and their ability to remove nitrate. So I used the char alone, and I also made duplicates where I had the chars by themselves, and then I used a 1-to-1 ratio of 0 valent iron with these chars. And so there was char and then char with 0 valent iron. What I did is I was able to put these in a nitrate solution and allow them to mix for 5 days. From there, I was able to filter the solutions to prevent any more reactivity. And then I took the visualization at 400 nanometers to see the amount of remaining nitrate in the solutions. So looking at the nitrate removal over time, 5 days was the best. So the longer that the nitrate and the biochar was able to react, the more nitrate was removed. Specifically, the most effective char was the biochar created at 400 degrees and maintained for 10 minutes, which also included the zero valent iron. In that char, we saw a 46% reduction in the amount of nitrate. That was a 46% reduction in the amount of nitrate over 5 days compared to that same exact char without zero valent iron, which had a 34% reduction. Then I looked at the effect of the heat treatment duration. So I looked at the 5-day period and I looked at the difference between chars held at the 0 time or the chars held at their temperature for 10 minutes. I was able to see the chars that were held for a longer duration were able to effectively remove more nitrate. And so this also compared with the chars at zero valent iron. There's a 12% increase in nitrate reduction by chars with zero valent iron compared to the char only. And the nitrate reduction increased by 8.2% when the pyrolysis temperature was held for 10 minutes as opposed to no time. I then looked and compared my char samples with 400 degrees overall with my 600 degree samples overall. And from there, I was able to see at each increment with 0 time, 10 minutes, with and without zero valent iron. Predominant influencer of this reactivity. So as the nitrate was reduced to ammonium, protons were removed from solution. And so with the removal of protons from solution, we'd expect to see an increase in pH to confirm that this was in fact reduction. And so at pH, at day 5, took pH data, and the pH data supports that in fact the reduction was the main source. So my 400 degree char with zero valent iron we saw was the most effective at removing nitrate. So in theory, it should have the highest level of pH increase. And in fact, it did. It increased by 2.25 pH. So now that I was able to see what the mechanism was and that I established a good char for removing nitrate, how I can use this in a real world situation. So the most direct method would be through a permeable reactive barrier with my pistachio biochar. And essentially what that is intercepting groundwater flow by digging a large column into the ground. That column can then be filled with biochar in the same way that a Brita filter works or your fridge filter works. As the contaminated water flows through the filter, it will, as we saw in the experiment, should be reduced and clean water will be coming out the other side. So there's lots of potential for this. And in the future, I would hope to see maybe a more optimized temperature range. I was able to see 400 and 600, but then playing around in the 400 range, I could find a potentially more effective remover of nitrate. Additionally, I focused on nitrate, but there are other capabilities of biochar. So heavy metal removal, other contaminants, it's a matter of how it can be modified and what temperature it is produced at. So while nitrate was seen as being successfully removed, that is not the only option, and there's still lots of potential for this technology. So thank you for listening, and I'd love to open it up for some questions.
Again, I apologize, but we're being asked to stay to a very strict schedule, and it's been emphasized several times, so I really apologize for not having time for any questions, but please be sure to contact Riley directly if you have questions, and we'll give our tech team a second to set up for the next speaker.
Thank you.
Thanks, Riley. Okay, so we now present Satvik Basareddy, who will present on near-infrared fluorescence imaging in mice with graphene quantum dots.
Hi guys, my name is Satvik Basareddy, and I'm here to present near-infrared fluorescence imaging in mice with graphene quantum dots. So to start, over the past several years, we've seen nanomaterials being used as drug and gene delivery vehicles, as well as imaging probes in various biological systems. So the reason why near-infrared imaging with nanomaterials is very useful is because imaging in this way allows for low biological autofluorescence, low tissue scattering, and it allows us to penetrate the animal tissue a lot more deeply. And this allows us to image the organs of the body more effectively. And current nanomaterial platforms are not very biocompatible. They're very difficult to prepare and they're very expensive to make as well. So for this reason, we developed graphene quantum dots, or GQDs, and we synthesized these from a microwave-assisted bottom-up approach, using glucosamine as a starting material. So as you can see in the schematic in figure A, we have glucosamine as the starting material. It's subject to a hydrothermal reaction in the microwave. And this creates the lattice structure that gives rise to graphene quantum dots. And in order to obtain the near-infrared emission from the quantum dots, we doped them, or we modified them with thulium 3+ ions. And these ions are really interesting because they contain intrinsic atomic transitions in the near-infrared region. So if you look at the far right on the figure, we can actually see the H4 to H6 atomic transition. And this transition allows for near-infrared emission from the quantum dots, which is very useful for imaging in mice. And another plus of our technology is that these GQDs are very biocompatible too. So after we made the quantum dots, we gave the samples to MIT, and they did the in vivo and ex vivo imaging study. So as you can see in figure A, we can see the emission from the quantum dots inside the mouse at various different time points. If we look at figure C, the ex vivo study, they extracted the organs, and we can actually see the emission of the quantum dots inside several different organs, such as the intestine, the kidney, the liver, and the spleen. So afterwards, we imaged the individual tissue slices from the 4 organs. We imaged them, we imaged the control that was untreated, and we imaged the tissues that were treated at several different time points, the 1, the 3, the 6, and the 24 hours. So the reason why we analyzed these organ tissues is to ensure that the GQD fluorescence actually exists inside the animal tissues. And the other reason we did this is to find the specific locations where the quantum dots were emitting light inside the tissue for imaging purposes. So for each bright spot inside the organ tissues, we took the fluorescence spectra, and we compared that fluorescence spectra to the bulk quantum dot fluorescence spectra from the sample itself. So as we see in the 6-hour condition, we see a strong peak at 940, whereas the control basically is a flat line. And this peak at 940 matches up with the fluorescence spectra from the bulk quantum dots, as shown in the right. So this confirms the identity that the quantum dots actually exist inside the tissue and that the fluorescence is real and it's not an artifact. We see this fluorescence at the other time points as well. We see a lot less in the 1 hour, a lot more in the 3 hour, and it tapers off with the 24-hour condition. And this implies that the fluorescence increases from 1 to 6 hours, but when it drops when you get to 24 hours because the quantum dots are being excreted from the animal, if that makes sense. And we see the same trend with the liver and the spleen as well, as shown in these figures. So to summarize, we believe that our doped GQDs are very promising for bioimaging probes because they're highly biodistributed, so we can see them in multiple different organs, and they emit near-infrared light in a broad range of targets. And since we can image live animals without dissecting them, we can reduce the number of animals sacrificed. And these results are promising for future image-guided drug delivery applications. And a possible modification, we didn't see any fluorescence from the brain, so maybe we could modify the quantum dots with other structures so they can cross the blood-brain barrier, and perhaps we could study neurodegenerative diseases. And these are my acknowledgements. And I'm happy to take some questions.
We do have time for a couple of questions. You can post it in the chat or raise your hand and we'll give you privileges to speak, ask your question. So I had a quick question. So would you expect, could you potentially use this for diseased tissue versus normal tissue? Is this something that might show up differently in a tumor per se, or something like that?
That's a great question. Yeah, we couldn't do tumors for this project. So I guess this is like a proof of concept, but the intended application for this is, let's just say you have a tumor inside an organ. You inject these quantum dots into an animal or an organism, right? You can use the near-infrared camera to illuminate the tumor location, if that makes sense. So it's like a diagnostic tool. And I would say that we actually found that you could conjugate these quantum dots with drugs. You can deliver them into an animal, so you can actually locate the tumor and treat it at the same time.
Thank you. I appreciate it. Thanks, Safik, for your presentation. We did have one more question in the chat, but I'll have her email you directly. That's okay.
Sure.
All right, so we now present Austin Bryant, who will speak on the effects of early life stage thyroid disruption on reproductive behaviors in fathead minnows.
Thank you for the introduction. So we're, hold on, I'm not in full screen mode. There we go. So we're interested in this topic because the previous lab student, Peter Bruns, found that early life stage thyroid disruption has some pretty interesting effects on reproduction. Peter exposed newly hatched fathead minnows to propylthiouracil, which is a model thyroid synthesis inhibitor, until about 40 days post-hatch, and then raised the fish to sexual maturity in clean water, and then evaluated their reproductive performance. So if you look to the graph, you'll see the reproductive performance of 4 different pairings of fish. And you'll notice that when the pair contained a PTU-exposed male, there was a significant decrease in fecundity, indicating that it was the exposure history of the males and not the females that leads to a decline in fecundity. So as a part of that study, it was also determined that the gonadosomatic index, sperm motility, and fertility success were not impacted by early life stage thyroid disruption. However, when another graduate student, Abby Johnson, conducted next generation sequencing analysis on the brains of these male fish, she found that those that were exposed to PTU experienced significant alterations in the expression of genes associated with neurogenesis. So we know that early life stage thyroid disruption alters the expression of genes related to neurogenesis, and that there is a subsequent loss of reproductive output. So now, given the relationship between neural development and behavior, we hypothesized that developmental thyroid disruption leads to altered reproductive behaviors. Therefore, the objective of this study was to determine if reproductive behaviors differ between control and PTU exposed males. To address this objective, fathead minnows were into a control group and a PTU exposed group. Exposures began just a few hours after hatch, and at 7 days post-hatch, a subset of fish were collected for confirmation of thyroid disruption via morphometric analysis, immunohistochemical labeling of thyroxine, and thyroid hormone-related gene expression. After 39 days, the exposure period ended, and all fish were moved to clean water and raised to sexual maturity, at which time I evaluated their reproductive behavior. So fathead minnow reproduction involves really intense bouts of behaviors, which are split into 2 main categories. The first being competition, where males will display aggressive behaviors to each other in order to gain and defend a nesting site. And the second is courtship and cleaning, where the male will clean the nest and display behaviors in an attempt to kind of lure her into the nesting site and spawn with her. To quantify these key reproductive behaviors, I set up tanks containing 2 males from opposing groups, a single control female, and a nesting site, which in this case is a split PVC pipe. I then recorded the behavior of the fish in 5-minute increments twice a day for 2 consecutive days. And so when I was analyzing the videos, I recorded the number of blatant behaviors performed by each male, specifically looking for headbutting, chasing, tail whipping, and the competition behaviors, and courtship, in the courtship behaviors, headbutting, lateral display, and spawning. And so here is a video of 2 males vigorously headbutting each other in an attempt to kind of gain and control that nesting site. So as you can see, it does involve, pretty intense, bouts of behavior. Sorry. In addition to quantifying specific behaviors, we also determined the winner of each trial, with the winner being the male that clearly displayed the greatest number of aggressive attacks. And we found that the PTU exposed fish were significantly more likely to lose than the control fish. In fact, the high PTU fish lost 82% of the trials against the controls. So this finding suggests that early life stage thyroid disruption leads to alterations in male competition behaviors. Been looking at all three of the broad categories of behaviors. We found that the exposed males significantly underperformed relative to the control males. PTU exposed males displayed a 71% reduction in competition behaviors, a 73% reduction in courtship behaviors, a 75% reduction in nest care behaviors, which resulted in a 73% reduction overall in behaviors. So this provides further evidence that male competition behaviors were altered among PTU exposed males, but it also demonstrates that courtship behaviors were also altered. The alterations in reproductive behavior that we saw may explain the lowered reproductive output that has been previously observed among fish exposed to PTU. Fish reproduction involves a complex set of behaviors aimed at acquiring a nest, courting a female, and keeping the nest clean. And we've seen alterations in behaviors that relate to these three aspects, which provides pretty strong support to back up the previously documented alterations in fecundity. I'd just like to thank everybody for coming out to my presentation. I'd like to acknowledge, my PI, Dr. Muller Jeffries, my committee members, Dr. Matt Hale, Dr. Gary Bohm, all the people in the TCU or in the Jeffries lab, especially Dalton Allen for helping me out. And obviously couldn't have done this this research without the the CERC grant funding and the Adkins Fellowship. So I don't know if I have time to answer questions, but if I don't, feel free to email me.
Thanks, Austin, for your presentation. And yes, we do have time for a couple of questions. Again, you can either post them in the chat and I will see them, or you can raise your hand and I will unmute you. So I did have a quick question, and you may have covered this, but the inability to compete, could that just be due to like change in mass or size? Like, did those early effects have changes in just basic, morphology?
Yeah, so that's a good question. So I size matched the males when I picked them and put them in the same tank, but I did it by eye, so I didn't always do like such a great job. So for any fish where there was a difference of 30% or more in the mass, we excluded it from the analysis. Because obviously, like the bigger fish will just typically always dominate the smaller fish, even if they've been exposed to PTU. So.
And you didn't see a difference in between the different kinds of competition behavior? You didn't see a difference there in how they were fighting or anything like that? Was it just a matter of they gave up sooner?
So typically, yeah, they like a lot of times they wouldn't like just display the behaviors at all. And like the control fish would like immediately like gain control of that nesting site, sometimes without even having to like display any behaviors. Like it would just swim straight forward, and the PTU exposed male would just swim around, not do much. So.
All right, well, thank you, Austin, for your talk.
Thank you, appreciate it.
And we currently have not identified our next speaker for the section. So if your name is something different than what's listed on the program, if you'll let us know. If we don't have our presenter for the next section, for whatever reason, could be a technical difficulty, then perhaps we can go back and I can actually, at least with Satvik, I can read the question from the chat, get an answer from you now. Oh, I do have a question from Gina, which says, this was very interesting, thank you. Has female behavior been evaluated with this exposure?
So typically when it comes to fathead minnow reproduction, it's driven heavily by male behaviors. But we did not address looking into the female behavior because I mentioned before that Peter's research, he found that it was the exposure history of the males and not the females that led to a loss in fecundity. So we were specifically focusing on male reproductive behaviors. Yeah, thank you, Gina.
So I received a message from someone indicating they were the speaker, but then I lost them. If you're back on or you're here, if you go ahead and send me another chat and let me know so we can make you a co-host and highlight you. All right, well, while we get that figured out, Sadvik, there was the question to you was, great job, Sadvik. What are some other medical conditions and diseases you think would be most promising for use of this? Oh, and I think we unmuted you at the same time, thereby canceling out each other.
Yeah. Yeah. So besides the more common classic example of tumor imaging and tumor diagnosis, I think another area that I think that we could use is, I think I briefly touched upon this, for neurodegenerative diseases. Those are very difficult to cure. I think since we can actually conjugate these quantum dots with drugs, we can transmit them through the blood-brain barrier. And I think a good area would be locating, plaques, right? Tau proteins right in the brain with the quantum dots, because we can image the near infrared emission from the quantum dots. So locating those plaques will be very useful, and we can also deliver those drugs over there, and hopefully maybe these diseases could be more treatable. I would hope.
Thank you. Okay, so I did receive word that the speaker for the 1:10 time slot will not be able to attend. So we will have a short break and we will pick back up at 1:18 with Jason Balters.
Hello, hello, testing. Can everybody hear me?
Yes, we'll hear you. Yes, we can hear you. And I will introduce you in less than a minute.
It sounds great. Jordan Pitts in this meeting is also my research partner, so we'll be co-presenting this presentation together.
Okay. Yeah, thank you for letting me know that.
Sure.
I will give him co-host abilities as well.
Hello, can you hear me? Okay, thank you.
All right, welcome back. We had a short recess. We had a speaker who was unable to make it. We are going to continue our presentations here at 2021 Virtual SRS. And we now present Jason Balters, who will speak on L-hoist North American dryer. If you have questions, you may raise your hand after the presentation or post them in the chat.
Okay, we will actually be speaking on determination of caffeine use and its effects on university students.
So hello, everyone. Thank you for coming to our presentation.
So some of the background of our research, we found a lot of conflicting outcomes when we did our literature review when it came to things like student habitual intake and sleep habits and the effects of caffeine use on stress. So our purpose statement was a key outcome of this research is to clarify contradictory results from previous studies on the topic of caffeine consumption, specifically amongst the TCU population.
The aims of our study were to determine how caffeine use affects sleep, perceived stress, modalities of caffeine consumption, quantities of caffeinated beverages consumed, and the perceived effects of caffeine withdrawal within the TCU student population.
So this research design was a cross-sectional observational study, and to complete this, we did a survey created on SurveyMonkey, had 17 questions, and we obtained written consent. Some of the different question types that we used were multiple choice, select all that apply, Likert scales, and agree or disagree. This was distributed to over 1,000 students via email, and this was over multiple different departments in the TCU faculty. And the data analysis we performed was through the SurveyMonkey statistical analysis software, and we did basic quantitative analysis and frequency statistics.
We had 217 respondents with 95% answering all questions. 77% was female, 72% were white, 16% selected Hispanic/Latino. We wanted to get demographic statistics, but it should be noted that this is not the actual representation of the TCU student population demographics.
Okay, so for one of our first questions, assessing caffeine intake, we assessed average number of caffeinated drinks per day, and we assumed one drink to mean 95 milligrams of caffeine because that's 1 cup of coffee's worth. Most students answered from 0 to 1 or 1 to 2. So together we had about 80% of the participants consuming 1 to 2 drinks per day. And then we only had about 1.8% of students consuming more than 4 drinks per day, which would be over the FDA recommendation to stick to less than 400 milligrams of caffeine intake per day.
This graph compares male and female modalities of caffeine consumed. Coffee and tea are the most popular drink choices. Men reported higher consumption for the energy drinks, natural energy, and caffeine supplements categories. These products are almost exclusively marketed towards men. And if we had more male representation in our study, we may have seen higher responses in those categories. One problem with the question for this graph was that we did not provide any answer choices for those that do not consume caffeine. So we would want to include that on a future survey.
Absolutely. So looking at some of the relationships between caffeine, sleep, and stress among the TCU students, most students reported a neutral effect between caffeine consumption and stress. So about 66% of the participants, or about 2/3, reported that their stress was not related to their caffeine intake. And about 50% of participants said that stress was rarely a trigger for caffeine consumption. Looking at some of the sleep trends, most students thought that caffeine had a neutral effect on their sleep quality overall, about 66% again. And then we did see most students often felt prolonged wakefulness, with 51% choosing that option.
The top three withdrawal symptoms reported were headaches, exhaustion, and fatigue. And the least reported symptoms were nausea, depression, and fever.
So our last question here was to choose the agreement with the following statement, and it was to get an idea of the perceived negative impacts of health expressed by TCU students. So the students chose their agreement with the statement: the effects of over-consuming caffeine would have a negative impact on a person's overall health. So a lot of students agreed or strongly agreed with this statement, about 78%. 14% were in the middle ground with neither agree nor disagree. And then we had an 8% combination of disagree and strongly disagree for this question.
The consumption of 95 to 190 milligrams of caffeine per day by the majority, which is 55% of the study participants, was consistent with the reported literature. Only 1.8% surveyed reported higher than the FDA upper limit of 400 milligrams of caffeine. And 70% of those who stated they experienced withdrawal symptoms, we actually don't know. We can't correlate that data to see if that is actually correlated with caffeine use. So further research regarding caffeine consumption by consumers is also warranted to better assist RDs and other health professionals when assessing diet and lifestyle habits.
So for our study limitations, we had a smaller sample size than we would have liked to see. A lot of the research in our background had about 500 participants. So in future samples, we might open this up to other universities so that we could get better numbers for our sample size. We also had a non-diverse sample with a lot more female respondents than male. We think we would have had a lot more people answering that they consume energy drinks if we had more male representation, as Jordan said on an earlier slide. And then we also had some limited answer choices and phrasing of questions that could have used some editing on a future survey. And for example, like on the question that assessed student intake on drinks per day, we would want to have more accurate numerical choices that reflect individual responses rather than overlapping ranges. That was problematic when we went to go do some of our data analysis.
Okay, applications for the RDN, registered dietitian nutritionist, are to know that coffee and tea are still the top two choices. It's rare for students to not consume caffeine, and you need to be aware that the upper limit of 400 milligrams per day exists, and be able to make your patients aware of caffeine intoxication and withdrawal. And that is our presentation.
Yeah, so these are our resources, our references. And if there's any questions, we'd like to open it up. I see we had something in the chat, but I didn't get to check. Just time.
Yeah, unfortunately, we are out of time. So feel free to email them directly with your questions. And thank you guys for your presentation.
Thank you.
Thank you very much.
And I apologize for announcing the incorrect title.
Oh, it's okay.
We'll take a couple seconds to switch over to our next presenter, and then we'll get going. All right, we now present Kyle Ruskey, and he will talk a little bit about L. Holst North American Dryer.
Yes, thank you for that. So my name is Kyle Ruskey, and I am representing the senior design group working for LWASP North America. Our team is a little bit unique. This year we are inheriting last year's group project, essentially meaning that we're actually taking over a project for the very first time in senior design history. So what exactly are we doing here? Well, we have we are tasked with building a rotary steel lab scale rotary steel dryer for LWASP. Essentially, the purpose of this is for LAWAS to be able to test certain variables for their limestone drying. The dryer is a steel cylinder. It's about 36 inches in diameter and 5 feet in length. The inside of it are covered in metal teeth, which we call lifters. The purpose of these lifters is to move the limestone through the cylinder and to get a veil, so to speak, of limestone falling down through the middle of the cylinder. We aren't actually drying this limestone, but if we were, it would be through air right through the middle of that cylinder. So when it was falling, that's when it would be dried, ideally. We can spin the cylinder up to 10 RPM. We can actually get it faster than that, but we're not supposed to do that. And the whole purpose of this is to give LAWAS something to test. Specifically, they want to be able to test the type of lifters, lifter spacing, the inclination of the dryer, anywhere from 0 to 5 degrees, as well as the dryer speed, or the RPM of this dryer. So before we could do that, as I said, we were inheriting this project from another group from a previous year. So we had to go in and re-verify some of their hand calculations, specifically for the supporting structure. If you saw on the previous slide, there is in fact a very large structure supporting this. We have to be able to support up to 3,000 pounds when factoring in our factor of safety. We need to make sure that it could do that. And what we found is that is 100% the case. It can go far over this, actually. It's very over-engineered. It is also made of mild steel. We also performed finite element analysis calculations through Inventor Nastran, just to ensure that there weren't any areas, high areas of stress where the structure might buckle. And so once we were able to verify that the structure was indeed safe and that we could in fact use it, we needed to find a way to actually affix the cylinder to this structure. And this is where we have our roller brackets. Now we were given these rollers and the axles for them from the previous group, but we needed to find a way to make it work. One of the issues is that the cylinder isn't perfectly round, and we needed to give ourselves a little bit of room in order to place these brackets perfectly where we needed to. So we designed the brackets with slots that were going horizontally, and we affixed this to the structure with slots that were going vertically. This would give us movement in both the X and Y direction. From this, we would be able to support, we would be able to have the dryer sit on in any position that we needed to, and we were sure that it was going to be secure. If we go to the next slide here, and the final part of this was to make sure that the dryer could spin correctly. And to do this, we had to hook up a VFD to our motor, and we, using a knob, we can now adjust the rate of the spins per minute, the RPM. We want to go anywhere between 5 and 10. That's what LAWAS has asked us to design to. We had to also install a tensioner, which you may see is that little bump. I'm not sure if you can see my mouse there, but that little bump right there next to the motor. This is to ensure that the belt stays tight and that it won't, that we can actually spin the thing exactly how we want it. And that is essentially the work that we have done. Is there any questions about the cylinder?
Yes, so we do have a question from the chat.
Oh, let me look at that. I cannot see
first
the chat.
question. Oh, no worries. Yeah.
Okay.
Yeah, I'll go ahead and say it. I was just letting you know.
Thank you.
where it's
Yeah,
coming from.
that's fine.
So how much force can the lifters themselves withstand? Also, what is VFD?
Yes. Okay. So for the first question, the lifters themselves, how much force can they withstand? I believe we have designed those lifters. Well, it depends on the lifter. There's multiple types. So for the first, one of the first types is a radial lifter. Let me see if I can actually show you all in a picture. Some of these lifters are just straight, that don't have like a bend to it. The limestone's supposed to fall off of those pretty quickly. You're not supposed to have too much weight on them. For the exact lift, the exact force per lifter, I'm not entirely sure. They are made out of mild steel and they are welded on, or in certain cases, some of them are welded on, so they can withstand quite a lot, but there's not supposed to be that much limestone per lifter at any single moment. So that really hasn't, we've never run into that issue. We even had cardboard in there at one point and they withstood that pressure. So they're not undergoing too much. And VFD stands for variable frequency drive, was that second question, I believe. Anything else?
So real quickly, this is a neat project because it sounds like the production side says, hey, we want something like this that we can test. And then you guys, how much freedom did you have in terms of, you know, they want this dryer made and that's pretty much all they give you, or do they give you more restrictions? And I'm curious what sorts of challenges you faced with trying to balance this is what they want with, you know, whatever limitations.
A big challenge and a big, I guess you could say that we endured was that by inheriting this project from another group, we were already set on a certain path. And so we had to make that path work, whether that's how we would have done it or not. The WASP was really good though. They provided us with lots of information. They made sure that we knew exactly where to go. They provided us with the cylinder itself. Essentially just the whole thing is like, hey, get this big thing on a structure and make it spin. That's kind of like the bottom line of it. We are also tasked with building a feed system. That will be a later presentation, not given by me, but they've helped us a lot with that and exactly what they want and whatnot. We are given room as to how we go about it, so long as we hit certain things. For example, they want to be able to put 1,500 pounds in this thing. So like that was like our main thing. But other than that, as long as it spins at the rate that they want it to, then they're pretty much cool with it.
Well, thank you. I appreciate that. So we now present Sachi Wirasaria, who will present on Starwell's Rise of the Satellites.
Thank you very much. So, hello everyone. Welcome. My name is Sachi Wirasaria. Welcome to my poster presentation, Starwell's Rise of Satellites. So this poster shows results from my recent work with my advisor, Dr. Mia Soderberg. So what is a star well? Star well is a gravitational potential well that encloses stars. So if a particular star has a gravitational potential lower than that of the galaxy, then it is bound, both of them are bound together and with that galaxy. So hence, galaxies act as star wells, collecting stars in their potential wells. So we study small and faint galaxies, but why? What makes these tiny galaxies special is that they're very sensitive to their internal and external environments. For example, even the smallest stars and the explosions of small stars and the stellar winds can expel gas and dust out from these galaxies, affecting how they form stars, and it's also going to affect their host environment, host galaxy. So in this project, we have used a multi-body simulation of a Milky Way-like galaxy to produce properties of a galaxy named the Large Magellanic Cloud, also known as the LMC, and its satellite galaxies surrounding it. So LMC is a large, small galaxy, small faint galaxy, which is close to our Milky Way galaxy. And the simulation that we use contains only dark matter particles. So they do not interact with light. But if we want to study stars, we need to introduce the star formation effect. So we introduce galactic physics and star formation physics by solving a bunch of differential equations. Now these equations involve gas cooling, forming stars, stellar explosions, and mass loss from these stars, as shown in the cycle. So in the figure, the figure 1, you can see this cycle. We call this the baryon cycle, which shows how stars are formed inside a galaxy. Now, then, once we do this, we compare the properties like chemical composition, light of each galaxies, and their half-life radii, and various other properties to observations. So on the right-hand side, you can see the chemical composition plotted as a function of absolute magnitude. So this absolute magnitude is a measure of brightness. So from going from left-hand side to right-hand side, the brightness increases. So you can see the brightest galaxy here in this figure is the LMC, which and the rest of it are galaxies within 512 light years within the LMC. And we predict the chemical composition and the brightness of LMC fairly well. And you can see the next brightest simulated galaxy, which is a possible small imaginary cloud, SMC, also has a good chemical composition, but it we underpredict its brightness. So we still have somewhere where to go to tune our model and calibrate our model further. So in the middle, in figure 2, you can see the distribution of satellites around the LMC galaxy and within 512 light years. And these points are, these satellites galaxies are sized by the half-light radii. This is the radius at which half of the light inside the galaxy is found, and they're colored by their brightness. So the lighter color you have, the more brighter they are. So in conclusion, we have produced the properties of satellite galaxies around Milky Way. Especially here, we are looking at the LMC galaxy and its satellite galaxies, and we have predicted the chemical abundance and brightness fairly well, but it still needs improvement, and we hope to achieve this in the future. And I'm happy to answer any questions.
And we do have time for questions. You can post them in the chat, or you can raise your hand and I'll call in. So what are the different parameters that are taken from the data, e.g., for example, spectrum, etc.?
So the parameters in the model, we can vary different parameters affecting, for example, star formation laws, different star formation laws, or how the efficiency of the stripping of stars from these galaxies and things like that. So there are different parameters in from like how much gas we are providing for the galaxy. So there are different things that we can vary.
And then what would sort of be the next step for your data?
So this was part of the side project that I wanted to just look at the LMC satellites there. So the next step is once we calibrate the Milky Way satellite galaxies, I would be looking at a different galaxy which is located further away from us and which is not explored much in detail, and predicting the properties of that galaxy is the next step in my research.
Well, thank you. Thank you for presenting.
Thank you.
Give our tech team a second to set up. And received an email from Kimon earlier, but I don't see him at the moment. If you're there, or if you're under a different name, shoot me a message in the chat just so I know who to highlight. If not, we'll give him a minute. All right, I see Kimon. So we will now hear from Kimon Vogt, and he will present on AI attendance and emotion detection.
Thank you so much.
And again, if you have questions, you can place them in the chat and I'll read them aloud, or you can raise your hand at the end of the presentation. And if we have time for questions, I will let you guys know.
Thank you so much, everyone, for coming today. My project is funded by CERC and my advisor is Paul May. I actually changed a little bit the title in order to in order to like be more accurate with where the project is right now, because the title was given when the project was still in progress. The project right now is heavily based in transfer learning. Therefore, the title is Attendance and Safe Classroom Access Using Transfer Learning. We was inspired to create this patent by a lot of reports of trespassing in schools and universities, and because of COVID. The patent will help professors take attendance before class and measure the temperature of the individual students in order to ensure safety. It will help prevent or decrease the COVID spread and other diseases that include fever. And it will also keep the students enrolled in the class and the professors safe. As you can see by the flowchart, we use the OpenCV library and the face recognition library. The OpenCV library is used in order to read the external camera's input, and the face recognition library is used through transfer learning to use a pre-trained model and fine-tuning it with the inputs of the students enrolled in the class. The model will do a facial recognition, and if it classifies that the student is enrolled in the class, it will ask the subject to come closer in order to capture its temperature. And if the temperature is below a threshold, then it will grant access to the classroom. Otherwise, if the person is unknown or the temperature is above the threshold, it will deny access and provide further assistance. As you can see in the primarily results, we have some figures. In the first figure, it's the model trained only in one of the two subjects that was in the research. And in the second figure is when the model was pre-trained in pictures of the two subjects. You can see that only the people enrolled in the class will be detected from the model. And in figure 3 and 4, you can see some limitations of the model, as in figure 3, we couldn't detect the second student with the face mask, even though the model was pre-trained with their picture. And in figure 4, the second subject, the first subject wasn't detected wearing a hoodie. So the research is still ongoing, and there is space for increasing the performance. But as by now, we will be asking the students to uncover their face in order to be detected by the model. In the further studies, we want to make the model more user-friendly and include also emotion detection during the class time in order to provide useful feedback to the professors about which part of the class time the students was confused or they was tired. And we also want to make modifications in order for the pattern to be more user-friendly. That's a quick introduction of my project. Thank you so much for your time. I think we have time for any questions.
Thank you, Kamon. And yes, we have a couple questions in the chat. So one is, how does the hardware determine the temperature? Is there some infrared data, or is it just measuring EM radiation?
So it's detecting it through an external device. I have an external device that is an infrared thermometer, and it's for the face, and then it sends the data to the computer.
And
it's still ongoing research, so this is not operated in the model yet, but it's in the further studies.
And then I have another question that says, would a facial recognition technology using LiDAR do a better and faster job of recognition?
Maybe, I don't know.
And then a follow-up from the original question was, do they have to hold it?
If they have to hold it,
I'm guessing the equipment, or
oh no, it's on the wall, so nobody have to hold it.
And then I had a quick question. So my son's school has something like this at the entrance. So when you walk in to pick up your kid, there's something like this. How does this differ from existing technology that's out there right now?
That's a good question. So far, I know until now, personally, I haven't seen something like that. So I'm not pretty sure about the school that you said that implement this, but I was we was like inspired by the thought about creating something like that. I have to like further like search about it. But as of in the best of my knowledge, I don't know a university that uses this kind of equipment. Exactly.
Yeah, I was also thinking, the ability to sort of detect 1 versus 2 people could be used as like a security function too, where if there's 2, then you don't allow access because there only has to be 1 person in the picture to make sure that each person is granted access.
Definitely.
But I'll have to take your question offline. I'll give our tech team a second to set up for the next presentation, and then we'll move on. Thank you very much, Simone, for your presentation.
Thank you so much. Have a good day.
You too.
Hello.
Hi. I'll giving our tech team a second to set up, and then I will introduce you here in a second.
Okay. Am I not sharing my screen?
Yes, you will share your screen.
Oh, okay.
You should have permission to do that.
Okay.
All right, so we'll now hear from Holly Gold, and she will be speaking on modeling the impacts of green infrastructure in the Village Creek watershed, Texas.
Hi, everyone. Thank you for coming to my presentation today. And again, my topic is modeling the impacts of green infrastructure in the Village Creek watershed located in Texas. So a little bit of background. So water is naturally transported through the earth and atmosphere through the hydrologic cycle. However, this can be influenced by natural and anthropogenic influences, one of which is urbanization. So in urban areas, high human activity and the replacement of natural land cover with impervious surfaces can lead to high pollutant discharge to water bodies and high runoff, which can cause flooding after storm events. So as you can see in these first 2 photos over here, we have a natural land cover and a highly urban land cover. And so during a rain event, stormwater, about 50% will infiltrate in a natural land cover compared to only 15% in an urban area. And then When it comes to runoff in a natural area, about 10% will become runoff, and then and 55 will in urban environments. And then in a natural land cover, about 40% will be released to the atmosphere, where compared to an urban environment, only about 30%. So you see a lot more remaining on the surface as runoff. And then in this photo right here, this is just a little cartoon graphic that shows just the various sources of pollution from urban environments and how they can really get funneled into our storm drains and released into our water bodies. So my study area is a watershed located in the Dallas-Fort Worth area of Texas, which the region in general is very rapidly urbanizing, but can come at the expense of the environment. So one approach to mitigate these urban effects are from the implementation of green infrastructure. So these are cost-effective, environmentally sound developments that can be used to collect and treat stormwater to reduce those effects of urbanization. So my objective is, again, to estimate the benefits of implementing green infrastructure practices on water quantity and quality in the Village Creek watershed. So specifically for water quantity, we're concerned with total discharge volumes, as they can bring high waters to neighboring areas. And then for water quality, we're mainly concerned with E. coli concentrations, because that is a problem in the watershed. So to do this, we will develop a SWOT model, and then we'll run different scenarios, some of with current conditions without any green infrastructure, and then some with green infrastructure practices implemented. And then we will take those and compare those two different conditions. So this is just a general setup of my methods. To start, we used elevation, slope, land use, soil type, and climate data to develop and really drive the model. And then from here, the model was calibrated and then validated for stream flow data. So that is our discharge. And then we collect data for pollution sources around the watershed. This can be from cattle to on-site sewage systems. And we take those pollution sources and input them into the model. And this is actually the step that we are currently at, just finishing up calibrating and validating the model for water quality. And then with this model, we will run the baseline scenarios. So the baseline scenarios will demonstrate the watershed's current water quantity and quality conditions. And then we'll run it again with the green infrastructure modules in place to simulate the new conditions. So then after all of the simulations are complete, we will evaluate the changes in discharge and then our E. coli concentrations from before and after the green infrastructure is implemented. So these are just some of the preliminary results. So these first graphs on the left show the pre-calibrated results for stream flow. So one way which we evaluated the model was through BFI, which is the base flow index. So BFI shows the total amount of discharge that is considered base flow. And so, total discharge makes up from base flow and then runoff. So we're trying to see how much is actually base flow. And so then the BFI for the observed stream flow is about 0.29. So that means about 29% of the total discharge is considered to be base flow. And then the pre-calibrated model had a BFI of 0.33. So in general, the model is over predicting the pre-calibrated model. And also you can see with these two graphs as well. So the shaded regions are the SWOT outputs. And then the lines are the observed stream flow data. So with almost every variable, it is over predicting overall. So the model, after calibration, the model showed to be excellent for simulating discharge in the watershed. And we looked at this through different statistical calculations and then also looking at that BFI again. So the BFI for the calibrated model was about 0.294, so almost exact to the observed. And so the statistics from this chart, it can essentially be interpreted that the model has a low uncertainty for prediction and a strong prediction strength for stream flow. And then also the model tends to over predict discharge in the calibration time periods, and then under predict by 0.4% in the validation periods. So far, so my conclusions and future work. So far, again, the model has shown to be excellent for simulating discharge in the watershed. And then water quality calibration is currently in the process of happening right now. So that's underway. And then once that is fully calibrated, we will run our different scenarios shortly to provide the results for how green infrastructure can really help with sustainable development in urban watersheds. And if anybody has any questions.
Thanks, Holly. Appreciate it. And unfortunately, we don't have time for questions right now, but if there was a question from Ken Richardson, and if you want to contact him directly or email him, it was a question about the database and where you get your data from. So thank you again for your presentation.
Thank you.
Our tech team is ready. We now present Cindy Argueta, and she will present on the impact of the COVID-19 pandemic on diet and health-related behaviors of adults. And you can share your screen.
I'm presenting with my co-researcher, Lauren Jackson. If you could make her a host, please.
Yes. Were you able to make her co-host? Okay,
Lauren.
You should be.
Oh, Lauren Jackson, not Laura.
She says she's unable to unmute herself. I don't think.
Okay, yeah. By coincidence, there was also a Laura Jackson. So I apologize. we gave the wrong person permissions. We'll get that fixed right away and you should be able to share screen.
Thank you.
Thank you.
All right, so I'm Cindy, and this is my co-researcher, Lauren Jackson. Our research advisor was Dr. Gina Hill, and our topic of our research looked at the impacts of the COVID-19 pandemic on diet and eating behaviors in the Tarrant County area.
Our purpose and research objectives are as follows. We wanted to identify factors that impact diet and behavior during the pandemic. We also wanted to determine how access to food has been affected during the pandemic and also assess changes in food waste due to the pandemic.
So starting off with a little bit of background, as of April 2020, there were around 700,000 Americans that lost their jobs due to the pandemic, resulting in a 4.4 unemployment rate. And although there wasn't a bunch of research at the moment looking at how the situation directly impacted diet and health, there were several studies that looked at certain nutrients like vitamin C, vitamin D, and zinc to be beneficial in the treatment of COVID-19. Studies also concluded that decreased consumption of inflammatory foods had beneficial effects against infection in the first place, which just goes to show how important of a role diet plays in health, even when it comes to COVID.
So we distributed an 8-minute survey and we had 52 questions. Those questions consisted of background, nutrition and eating habits, access to food, food waste, and demographics. We shared it with a local Facebook group, and we also shared it with the Tarrant Area Food Bank, and they shared it with their partner agencies. So now we're going to get into some results that we found. So in general, our population was majority non-Hispanic white females, and of note, 17% of our population was of Hispanic origin. The average age was 41 years, and the average BMI was 29, which is in the overweight category, and the majority of participants had a bachelor's degree or higher. The majority of participants gained weight that they did not want to gain. And of those participants that did not want to gain weight, they gained an average of 13 pounds. And on the other hand, people who did want to lose weight, they lost an average of 14 pounds.
So we also looked at alcohol and snacking behaviors and how those were impacted during the pandemic. And we can see that 37% of participants reported increased alcohol intake. And also with snacking, 60% of participants reported increased snacking consumption due to the pandemic. We looked at stress as well, asking if they feel more stressed now than they did before the pandemic began, which over 60% of participants agree with that statement. And then also we looked at certain factors in terms of gross annual income. So the lighter color represents those that earn less than $50,000 a year, and the darker color represents those that earned more than $150,000 a year. So according to the chart, we see that those in the lower income category were more likely to be unable to afford healthy food, more likely to be overweight or obese, more likely to use free resources to obtain their food, and they were also more likely to try and waste less food now due to the pandemic. And then taking a closer look at food waste, we see on the left that at least half of participants report that they do not throw away more food now. And then on the right, we see at least half of participants try to waste less food now than before the pandemic began, which just shows that it seems that food waste seems to have been decreasing due to the pandemic in at least half of the participants.
Our research revealed the following as statistically significant. So we found that the higher the household income, the less weight people gained, and the more ability they had to afford healthy foods. We saw that the more people were stressed, the more alcohol they consumed.
We also saw that participants who said they snack more felt they were physically healthier before the pandemic began. And then finally, those who believe their weight is not healthier now than it was before the pandemic began were more likely to have reported weight gain. So in conclusion, we determined that income level played a significant role in food waste, ability to afford healthy food, weight status, and use of free resources. And we also concluded that the pandemic led to increased stress levels.
We also found that stress was associated with increased alcohol usage and snacking, and we found that more than half of the sample gained unwanted weight gain. And that is all of our presentation. And if you have any questions, please send them to the chat. Thank you.
Thank you, Lauren and Cindy. We do have a couple of minutes for questions. So we have one from the chat, which says, do you know if soft drink use has changed?
I'm sorry, can you repeat your question?
Do you know if soft drink use has changed?
We don't ask specific questions regarding soft drinks. That would have been pretty interesting to see. I mean, I would assume since, being home all day, temptation is there.
So I was curious whether you would interpret the not wasting food. Would you interpret that as a positive or a negative? Because on one hand, it's good that people aren't wasting food. On the other hand, are they eating expired food or food they wouldn't normally eat? How do you interpret that data?
You go ahead.
I would interpret that that's a great thing that people aren't wasting the food that they eat, because around 30 to 40% of food is wasted. So I think in a time of crisis, we need to make sure that we're eating all the food that we buy.
All right. Well, thank you guys, and thank you to all of our speakers and all of our attendees. And that concludes this session. So thank
you.